A little applause won't hurt while he's climbing the stage. Thank you very much. Thanks, Sriram.
Great. Thank you. Thanks to Sid for this invitation to come and speak to you. So I'm at UCLA. My appointments are in computer science, human genetics, and this new department which we started. It's actually, the name was just announced a couple of weeks back. It's called the Department of Computational Medicine. And this sits right at the core of my research interest, which is how do we actually bring techniques from computer science and statistics to bear on problems at the intersection of biology and medicine. And I'll tell you a little bit about some of the projects and some of the experiences that we've gleaned from trying to apply machine learning and statistics in this domain. Next slide. All right, so one of the main things that drives me is in trying to understand the genome, the human genome. It's this sequence of about 3 billion characters, ACGT. All of us carry this piece of human genome in us, but with small modifications. So if you compare my genome to yours, we'll differ in about 1 in 1,000 positions. And the question is, why are these differences there? What impact do they have? And what can we learn from these differences? Next slide. So for example, we have a lot of reason to think that the reason why some of us are at higher risk for some diseases than others has to do with differences in our genomes. So we'd like to go from this 3 billion long sequence to try to pinpoint what are these changes that give us higher risk for some disease. Next slide. So it's really great to have this compelling problem. The question is, where does machine learning and statistics fit in? And for that, you need to have really large amounts of data. And what has happened in this field over the last 10 to 20 years is something called the genome revolution. So on the left here, you have this plot that shows the cost of sequencing or reading your DNA over some time. There we go. So the graph on the left is cost of DNA as a function of time. That was the previous one, yes. And you see that line here. That line is what is called, what we know as Moore's law. So Moore's law says the cost of computing halves every 18 months. And the blue line shows the cost of DNA sequencing. So starting at around 2001 was when the human genome was first sequenced. You see that the cost is decreasing at the rate of Moore's law. And somewhere around 2007, it's actually falling faster than Moore's law. So we are generating sequences faster than the rate at which we can actually analyze them. So now we are in a field where there is way more data. It's no longer data limited. And the question is, how do we actually make sense of this data? So another way of seeing the same thing is, in 2001, we have the first human genome. So this was a project that took about 10 years to complete, several billions of dollars. Fast forward to 2010, we now have data sets that you can publicly go and download with thousands of genomes sequenced. And next slide. And today we have data sets that are available with millions of genomes, in addition to the fact that it's not just genomes, but also availability of things like what disease risk these people carry. So we actually have data today that can potentially tell us about the ability to understand what genes cause certain diseases. We also have companies now, so this is not just happening in a research institution. Oh.
The distance could be a problem with works. Try. The next slide.
Let's try. So we also have companies. These are what are called personal genomics companies. So you have companies like 23andMe, where if you pay a couple of $100, you spit into a small test tube, they will actually tell you what are these genetic mutations that you carry. And then they will also give you some interpretation. So they will say, you have a mutation that increases your risk for a certain disease. So do I need to point there? Or here? Okay. All right, so I'd like to talk a little bit about what are the kinds of machine learning and statistical challenges that arise when we try to analyze and make sense of this data. So we'd like to actually go about building realistic models of biology, which go all the way from your genome to figuring out what traits you carry. And so one thing about biological data is even though we have millions of genomes, it's fundamentally the data is not big. The data is wide. So in other words, the data is high dimensional. There are more features than the number of samples that we can hope to collect. It's heterogeneous, so we're going to talk about different kinds of modalities. So Sid talked about the kinds of data that we're collecting in UCLA, and these data come with different kinds of biases. It's extremely noisy, a lot of missing data. And so when we actually want to build models, we have to deal with all of these issues. Beyond that, in biology, we don't just care about building models that have high predictive accuracy. We want these models to make sense in terms of biology. So we need models that are interpretable. Then the big challenge is we might build a model that is highly realistic. How do we actually get it to scale to the millions of genomes that we're computing? So this poses an inherent tension. You want realistic models, but then you want to actually make inferences at scale. So this is where algorithms and computing come into the picture. And finally, I'll touch a little bit about this, is in all of this data, there are huge concerns about privacy. So what can we learn about you that you did not really want to know about? And so how do we deal with these constraints? So how do we make these kinds of inferences while still making sure people's privacy is respected? All right, so in the first part, I'll talk about machine learning applied to genomic data. And just to be concrete, I'll talk about one specific problem, which gives you some of the ideas about the kinds of issues that we need to deal with when we're dealing with these kinds of data. So our goal is to interpret our genome. So we'd like to go from this 3 billion long sequence to being able to say, you carry a mutation that increases your risk for some disease. So one way to do this, and that's been a really powerful way, is a way which is completely blind to anything we know about biology. Next slide. So in this paradigm, we're going to collect genomes from people who have disease and from healthy people. So every row here is the genome sequence of an individual. And all we're going to do is going to go along their genomes and compare mutations across people who are healthy to people who have a disease. So for example, we're going to look at this column. And in this column, there are some people who carry the letter G and other people who carry the letter A. The technical term for this is what's called a SNP. And we're going to look for SNPs which are different in these two classes of individuals. So for example, if you look at the column on the right, it looks like both people who have disease and who are normal carry both A and G. So that's not very interesting. On the other hand, when we look at the column on the left, next slide, we'll see that this seems to be somehow associated with the fact that there is a disease. So you might look at this column and say, aha, we have found a mutation that is correlated with whether or not you have a disease. So what's the catch? The catch is, let's say I tell you that accidentally, all the people who have the disease come from one class of populations, and all the people who are normal come from a different class. So next slide. So let's say we just call them the blue and the red population, and this was not known to you when you actually collected these individuals. So having told you this, now this SNP no longer looks interesting, right? So this could simply be the fact that we have collected people who are living in two different places, and so they carry a mutation completely by chance, and this mutation correlates with the fact that there is a disease status. So in this case, this color, whether or not you belong to blue or red, is what's called a confounder. So if I were to tell you the color of an individual, it completely explains away this association between mutation and disease. Now what's the catch? The catch is often we don't know what these confounders are. So if you knew them, we would collect them. If, for example, I knew I should ask the person about the color, I would actually ask this in my study. The problem is, for most studies, we don't know what these confounders are. And in these specific studies, these genome-wide association studies, turns out a big confounder is your ancestry. So where do people come from? So imagine a study where all the diseased people came from one city, and all the people who are healthy came from a different city. So these people, just because of the fact that they've lived in different environments, are going to have different kinds of genetic mutations, different disease patterns. Next slide. All right, so we know that Ancestry is a potential confounder, so maybe let's ask people about their ancestries. Maybe that'll solve the problem. Turns out, that's not easy as well. So here is an example. So this was a study that happened 10 years back. What we did, or what this study did, was took genomes of a bunch of different people from all over the world. So there were about 1,000 genomes from different parts of the world. So there were genomes from Africa, from Europe, Central Asia, East Asia, and so on. So you just take the genetic data and you run some kind of clustering algorithm. So you can run different kinds of clustering algorithms. And what you see here is every vertical line tells you which cluster was this individual assigned to. So a lot of it makes sense. The first thing you see is, based on your genome, we can kind of predict your ancestry. We can say where you came from. So there's this red cluster, that's people who have come from Africa. There's this orange cluster for people who have come from East Asia, and so on. That's nice. So from your genome, we can infer your ancestry. But the picture is actually quite complicated. If you look at the Middle Eastern individuals, they have ancestries that both belong to Europe, Central Asia, and this new ancestry that's not found anywhere. And even if you take countries like Europe, there is a lot of variation in the ancestries. Next slide. So the bottom line is, ancestry is actually quite complex. And there are lots of individuals whose ancestries don't fall into any one particular group. And so this is what we call an admixed individual. And it turns out, as we get more and more genomes, we are realizing that most individuals and most populations are admixed. It's very rare that you find somebody who's completely orange or red. Everybody has bits of different colors in them. So what does it mean for somebody to be admixed? Next slide. Basically, the cartoon is something like this. There was some population that existed, say, 10,000 years back. It's split into two populations. Say one population went into Africa, another population went into Europe. And then later, these two populations come back together and mix. So what that means is nobody belongs to population 1 or population 2. They're going to be a mixture with different amounts of ancestries belonging to these different populations. Next slide. So the goal is we have to actually figure out what a person's population is from their genetics. And then we have to use that information to actually do disease association studies. So this is what it means to infer hidden structure. This red or blue is not given to you. You need to actually infer it from data. So you need statistical models for that. And then you take that and then run your disease association studies to get valid results. So to do this, we have to understand a little bit more about the biology. I'll tell you a little bit. The details are not too important. And the reason I want to say this is to show you how an understanding of the domain is extremely important in a lot of these problems. So the first step when we're actually writing a statistical model is to be able to figure out what are the aspects of this problem that are important, what are things we can ignore, and build it into our model. So we want to model an admixed individual's genome. So what does that mean? So here is a cartoon. We have people who are the ancestors of this population. So at some point in time, there are people who are blue or red, and they give rise to offspring, who give rise to offspring, and so on. And what happens is, because of the way your genome is transmitted from parent to child, there's this process called recombination, which shuffles up what gets transmitted. So if you look at the people who are the descendants, their genomes are no longer blue or red. They have this patchwork, where parts of your genome come from the red population, parts come from the blue population. So this is what it means to be admixed. You're never completely blue or red. Different parts of your genome might have red or blue ancestry. And this is something that we call local ancestry. It's local because it changes as you move along the person's genome. So this leads us to our first statistical problem. Next slide. This is our input data. So this is the data that we're collecting. Every row is an individual. Every column is the SNP. Again, we are computer scientists, so we don't want to deal with ACs, Gs, and Ts. We'll binarize everything. And so now we represent everything as a matrix of 0, 1, 2, Y0, 1, 2, because you have two copies of the genome, and you just count how many mutations you carry at every position. So this is our input, and we'd like to be able to infer this local ancestry for every individual. So what does that mean? We'd like to go and color each individual's genome according to red or blue, depending on the ancestry that they carry. So now there are many ways of solving this, but as I mentioned, there are a couple of criteria we want to have. One is the model should be interpretable. Second, it should allow us to scale to large data sets. So the kind of model that we're going to talk about here, it is not a deep model. It's a very traditional generative probabilistic model. What does that mean? It's basically a model where if I give you some parameters, things which you don't know about, and let's pretend you're God, you can take this model and have a recipe for generating data that looks like this. Why is that useful? It's not. What we actually care about is the inverse problem. Right? We don't care about generating fake data. We want to actually take data and figure out what were the parameters that were not known to us. But it turns out that's what statistical theory allows us to do. So once you've written down this generative model, there are tools that allow you to crank and go in the inverse direction. So I'll just walk you quickly through what this generative model looks like. Next slide. So it's what's called a graphical model. I won't say anything about graphical model because Raghu has a whole talk all about graphical models. So the basic idea is you write down all of the random variables in your problem. So here we have a whole bunch of random variables. There are W's, Z's, and X's. The Z's are the quantities that we want to infer, our local ancestries. The X's are the genotypes, this matrix of 0, 1, and 2. And here we have n of them because we are observing n SNPs for a single individual. And then we join them up. So we have edges connecting up these different variables, and these intuitively they capture the dependencies across these variables. So for example, if there's an edge going from Z1 to Z2, that's telling us the distribution of Z2 depends on Z1. And in fact, there's more. The lack of edges tell us about conditional independencies. So given this model, now let's write down a recipe for actually generating fake data from this model. So first we start with these W variables. Next slide. So these W variables are basically indicators. They're zeros or ones. And we set them zeros or ones independently according to some biological parameters. Again, details don't matter too much. Basically, what these W variables tell us is, was there a recombination between a pair of SNPs? So if the W2 is 0, that means the pair of SNPs adjacent to it have no recombination, which means they have the same value. If there was a recombination, then they can take on different values. Again, encoding some of the biology. So now you can set them to zeros and ones according to some parameters. Having set these W variables, now we're going to set the Z's conditioned on the values of the W's. That's why you have edges going from the W's down to the Z's. So next slide. The Z's are these local ancestries. They are blue or red. So we pick the values of these Z's while respecting the distributions that they satisfy. For example, W2 in our fake data is 0. That means there was no recombination. What that means is the local ancestry in the first and the second SNP have to be the same. In this case, they're red. On the other hand, W4, that variable is 1, and that is telling us those local ancestries can be different. So here you can have a switch from red to blue. So you can go ahead and set your Z variables according to this distribution. Again, there are parameters that control this which have some biological meaning. Next slide. Finally, given the local ancestries, we generate our genotypes, and again, there are some parameters associated with them. So at the end of this process, we have actually generated fake data, and now we can go to the problem we actually care about, which is inference. Next slide. So actually, we don't get to see any of the W's or Z's. They're hidden. That's what we care about. We only see the X's, the genotypes, and the problem is we want to infer W and Z given the X's. Next slide. So that's the inference problem. There are lots of ways you can pose it. In this case, we want to find values of W and Z that maximizes the posterior probability given data and some parameters. Turns out it's an optimization problem. There are lots of ways that you can solve it. In this kind of generative probabilistic model, a very standard way of doing this is something called an EM algorithm, which is a hill climbing algorithm for computing these kinds of posterior probabilities. There are subtleties in doing this. It turns out it's non-convex. There are lots of values of W's and Z's that can actually lead to a maximum, so you have to be very careful about initializations. And then finally, one thing I'll not talk about, but is important, is how do you do it at scale. So all of these are technical issues I'm happy to talk about offline. But let's say we've actually solved all of this. Now let's look at some results. So we have validated the model, and then we go and apply it to data. So this is some data given to us from a collaborator. So it's a study of asthma in these populations, in populations from Mexico and Puerto Rico who have very high rates of asthma, especially in Puerto Rico. We ran the algorithm. Here is one way of looking at the results. So it turns out these populations have 3 ancestry components. They have a component from Europe, another from Africa, another from the Native American populations. And what we can compute is, for each person's genome, what proportion of your ancestry comes from each of these 3 components. We see some differences. For example, the Puerto Ricans have more African ancestry, less Native American ancestry compared to the Mexicans. But even within each of these populations, you see differences depending on where they come from. For example, depending on whether you're a Mexican from New York City versus a Mexican from Chicago, the genetics can actually tell you these subtle differences. So this is not obvious if I just gone and asked somebody, are you Mexican versus Puerto Rican? Next slide. So now we've actually inferred hidden structure. So now we can actually go back to the problem which motivated this, which is how do you actually use this to study disease genes? There are lots of ways of doing this. So one idea is something called admixture mapping. Simple idea again. Look at people who have diseases and healthy individuals, and for every position along the genome, ask what fraction of people have African, European, or Native American ancestry at that position. If a position has nothing to do with the disease, it should be equal in cases or controls. If it has something to do with the disease, you will see a difference. So you can run this, and let's say you run this on the asthma cohort. We get something that looks like this. Next slide. So this is basically showing these differences in a statistical scale. And these peaks are telling you places where there's a huge deviation in what you see in cases versus controls. So in this case, there is this position along the genome, which seems like it's highly associated with asthma. It turns out this has something to do with their Native American ancestry. Turns out it's a very powerful approach. Next slide. And there are hundreds of labs that have used these kinds of techniques to localize genes for a whole bunch of other diseases. So cancers, which are very different across different populations, type 2 diabetes, and so on. Next slide. Here's another application of using ancestry in personalizing treatment. So this was a study that looked at children who had this very aggressive kind of cancer. It's called acute lymphoblastic leukemia. Turns out the recommended treatment is you have to do multiple rounds of chemotherapy. But it turns out not everybody responds well to chemotherapy. Some people have relapse. And so this study asked, how likely are you to get a risk of relapse depending on your ancestry? So they ran this method, and they looked at one kind of ancestry, which is Native American ancestry. And to cut to the chase, it turns out if you have Native American ancestry less than 10%, you are at a lower risk of relapse compared to if you have high Native American ancestry. So this is a setting where knowing somebody's ancestry would actually influence what kind of treatment you would assign to them. Next slide. All right, so just to summarize the first part, I talked about genetic ancestry and how it can be used for disease gene localization. Next slide. And how that most human populations are admixed across human evolution. And there are these new data sources, our ability to collect genome sequences, that can enable how we can infer this ancestry. And there are several methodological challenges that I can talk about offline about how these methods need to be adapted to the kinds of questions that we are now beginning to answer. All right. So now I'll quickly talk about the second part, which is the challenges that arise when we're looking at clinical data sets. Next slide. So at UCLA, we have a fantastic hospital system, and this has been the start of a very fruitful collaboration with clinics who are trying to apply machine learning to improve patient care. So I want to talk about one specific project that's quite far along. The goal here is to understand what leads to complications after surgery. So in the UCLA hospitals, there are about 70,000 surgeries that are done every year. There are a small percentage of complications. There are 1,000 people who die in the hospital after surgery. It's fairly small, but you'd like it to be even smaller. There are individuals who develop sepsis, infections. around 4,000 people actually develop acute kidney failure from these surgeries. So in all of this, we'd like to identify early on what are the risk factors and how this can be managed. Next slide. So what we'd like to do is, given information about an individual that is available before you go into surgery, you'd like to be able to predict what is your risk for one of these complications. So for example, what is your risk for dying in the hospital? And the reason we want to do this is, right now we are not fully at a level where we can automate these predictions. But the key reason is we'd like to be able to focus the clinician's time on those patients who are at high risk. Right now, the clinician spends all of their time equally across different patients. And secondly, we'd like to be able to monitor how this risk changes across time as the patient comes in, gets treated, and tries to recover. And so this is all in collaboration with the anesthesiology department at the UCLA hospital. So these are the folks who see a vast majority of the crucial surgeries that come in. Next slide. So this is how we actually selected our patients. So the patients are people who have what we call electronic medical records. We have electronic medical records going back 5 years. And so we start with about 300,000 surgeries. And we apply a bunch of criteria depending on how serious the surgery is, whether the patient was too old, too young, whether they came to one of the hospitals where they were followed throughout the surgery, and so on, till we come to about 60,000 surgeries. So that's our data set that we're going to be working with. Next slide. We extracted a number of features from the electronic medical records of the patient. It turns out the electronic medical record is extremely extensive. We have used only a small percentage of the information available. So this includes information like the general health, so age, BMI, weight, and so on, vital signs, blood pressure, blood oxygen, surgery-specific indicators, for example, what kind of surgery the patient is undergoing, labs, so the blood sugar levels, and so on. Next slide. So once we have all of this data, we ran several different models. We tried linear and nonlinear models. It turns out the nonlinear models as a whole outperformed the linear models. Amongst the different nonlinear models, we didn't find any statistically significant differences. So for now, I'll focus on results from applying a random forest algorithm. So everything we've done so far is mostly standard ML. And so a lot of the challenge has come in how we actually do the data processing, the data cleaning, and the interpretation. Next slide. Some challenges. So nothing to do with technical ML, but this is important to actually point out. I think a lot of this is typical of many data science and data analysis pipelines. First is defining your outcome. Believe it or not, it's actually challenging to figure out if somebody actually died in the hospital. So even though you have medical records, it's actually quite a bit of work on the part of the clinicians to figure out if somebody actually died. And so we spent a lot of effort. There's a huge amount of missingness. So we have to do some kind of imputation. And that leads to certain biases. So we have to evaluate the impact of those kinds of biases. There's class imbalance. So mortality is only about 2%, which is great. But again, that leads to issues of how do you actually evaluate these methods in a way that reflects class imbalance. Next slide. So now these are the results from running this classifier. So the nice thing about the hospital and the way this is set up is we are collecting patients as they come in. So we have patient records up to February of 2018. That's our training data. And then every patient that came in after that who was not part of our training data formed our test. So there's a very clean separation between training and test. And we are basically evaluating this algorithm in predicting the future. So it is a realistic way of measuring classifier performance. So the orange line is the machine learning, the random forest. And the things we compared it to were other clinical risk scores that have been used. So people have been using risk scores in the clinic. They're not very specific to the kind of surgery. They're not as extensive, but they're still widely used by surgeons. So we have 3 different clinical risk scores. And what you find is no matter whether you look at area under the ROC curve or the precision recall curve, the random forest is doing better. Turns out, for these kinds of class imbalanced problems, the precision recall curve is much more informative than the AUC curve. And so you see a much bigger difference when you apply the precision recall area under the precision recall curve to compare these methods. Another way of looking at whether this algorithm is clinically useful, so AUC is great from a machine learning point of view, so is precision recall, may not really tell the clinician whether this algorithm is useful. So here's another way of measuring this. On the y-axis, we are measuring the number of mortalities that this algorithm sees, assuming that everyone can be treated. The x-axis is how many patients had to be seen to detect a certain number of mortalities. So you want to see as few patients to be able to detect the same number of mortality. So what you see is, let's say you want to detect 20 mortalities, then the random forest sees maybe 20 to 10 times fewer patients. So this is useful because what it's telling the clinician is this is not going to produce false alerts that is going to waste their time. Next slide. Another thing we can do with this is all using information before surgery. Now we can do the same prediction using information after surgery. And now we can ask, how has the risk changed pre and post surgery? So what you see on the left here, on the diagonal, below the diagonal is what percentile of risk is a patient in before surgery. So high percentile means high risk, and above diagonal is percentile of risk after surgery. So generally, most people lie along the diagonal, so that's expected. You don't expect things to change much. But then you see certain interesting cases, people who have high risk pre-surgery, whose risk goes down afterwards, and the other way around. And the graph on the right shows you what happens to those patients who died in terms of their risk. And the interesting class of patients are people here. So people whose pre-surgery risk was high, whose post-surgery risk was low, but who still died. So what's going on there? And then, of course, you have classes of patients here. We did not pick them up pre-surgery, but who had high post-surgery risks and then died. So now this is useful because now the surgeons are going to go back, go to their charts and see what was the reason that the algorithm failed or what was the reason that the algorithm worked. Next slide. So that's just one outcome. Now we have ongoing projects in trying to do similar things for other kinds of complications. So here is another project that's in early stages where we're trying to predict kidney injury. And in this case, the machine learning method that seems to be doing well is some kind of a deep neural net. So compared to random forests right here and gradient boosted decision trees, the neural net seems to be doing much better in terms of area under the precision recall curve. Next slide. So just to summarize, I think there's amazing amount of rich signal in the medical records from the patient. And we've just started looking at it. We've looked at only about 50 features so far. The actual medical records have of the order of tens of thousands of features. And right now, there's a huge effort to collect multiple kinds of data sources. So we have labs, we have medications, we have treatment and diagnosis codes. What's coming down the pipeline is we're collecting waveform data. So in real time, we can measure blood pressure, ECG, EKG, and so on. Genomics, so everything I talked about in the first part. We're also collecting genome sequences on a subset of these patients. We're also collecting epigenetics, which measures dynamics. So for example, let's say a person smokes, there are changes to your genome that can be detected. Then of course there's imaging data, and that's of course a huge data which we haven't yet exploited. That said, all of this is at the level of the data, previous slide, but there are still lots of technical problems which need to be handled. I started out by talking about interpretability. The methods that do the best in terms of predictive power are these nonlinear models, these deep methods, random forests. But again, the clinicians come back and they want to know what was the one feature or handful of features that they need to focus on. So how do you go back and bake interpretability into these models? So when we talked about graphical models, that's nice because you get interpretability for free. The question of interpretability in these kinds of deep or nonlinear models is an open research question. Another major issue is how do you actually combine data from multiple different hospitals? So it's great that at UCLA we're sitting on this treasure trove of data, but there are other hospitals who also want to do similar things. And ultimately we want to combine data across these different sources. The problem is nobody wants to share data for obvious reasons of privacy. So how do you actually learn models while protecting patient privacy? So that's another major research question. Next slide. So hopefully I've given you a little bit of a glimpse of the kinds of problems facing a data scientist, a machine learning, or a statistician in the field of biology and medicine. There's a huge number of open challenges, both technical and applied. They're quite unique, I think, to these kinds of data. It's highly high dimensional, it's heterogeneous, missingness, noise, and sampling bias are major issues. We're not just interested in accuracy, we care about interpreting and putting these in the context of known biology. Scalable inference, I haven't touched upon this. So how do you actually do these on millions of genomes? That's a non-trivial problem. And doing this while respecting privacy. The other major thing I want to say is all of this requires close collaboration with domain experts to understand what are the key problems, to figure out what kinds of models and algorithms to choose, and finally, how do we translate these findings back into the clinic. Next slide. All right, so as I said, this is all a huge collaboration, both within UCLA and outside, and I'd like to thank our students, our collaborators, especially our clinical collaborators, and I'm happy to take questions.
Yeah. Hello? Yeah, regarding the first part of your talk, isn't it the case that you only looked at positive samples? Because there's like wide, population out there which has the same gene slices, but they haven't developed the disease. So is that the case or not?
So you're talking about the first part?
Yeah, the disease localization.
Right, so these are study designs. So there are two kinds of study designs you could do. One is you take a random sample in the population and wait for people to develop disease. The alternate is you look at people who have disease and then look at their genomes. Turns out they have different strengths. A lot of these studies are the second, where you've enriched for people who have the disease. And so you have to be very careful about how you translate these findings into the general population. And now there are actual data sets which allow you to track people longitudinally over time. It's just that you need much bigger sample sizes to get the same kind of power. But that's a great question. And again, so again, the key thing I want to emphasize here is the study design and how the data was collected is crucial in how you interpret a lot of these findings.
Hello. Hi, I'm Sriram, I'm CEO of Curl Analytics. We are into genomics. We work on HLA typing. I just wanted to understand when you showed about ancestry and how do you decode ancestry and so on, which part of human genome has that information? Like, most of the human genome has repetitive information. So what is that key region which you use for inferring these things?
Yeah, so there is this question of whether it makes sense to target a specific portion of the genome or to look genome-wide. And so everything we have done so far is genome-wide. So you're looking at about 500,000 to 1 million SNPs that are scattered along the genome. And there's a fair bit of evidence that that is the most accurate way to do ancestry inference, rather than look at specific regions. And a reason for that is specific regions might be driven by things like selection, which can bias these ancestry inference methods, whereas doing it genome-wide gives you unbiased estimates for a lot of these genome-wide parameters that we care about.
I think like you have already answered, but how do you figure out whether there's a mutation, like there are differences, whether they are because of ancestry or they are because of the disease? So.
You're asking how do we know whether a difference is caused because of difference in ancestry versus differences in diseases? Yeah, so this is why the genome-wide approach is useful. So our null hypothesis is there are a small number of regions in the genome that are going to be associated with the disease. And on the other hand, if ancestry is the reason, then you will see many regions scattered across the genome that crop up as showing differences. So this is how you kind of deconvolve whether what you're seeing is a null signal or whether it's actually driven by a biological result. So the idea is you don't expect 1,000 positions in the genome to all be associated with the disease. You could, but from a biological point of view, that's not an interesting result.
So, just asking further. So you would have figured out earlier that this region is responsible for this disease or
so let's say you actually run a scan and you ignore ancestry. Then what you're going to see is basically every position in your genome is going to show up as being associated with the disease. And you can do statistics, but the basic idea is, let's say you find that all million positions in the genome show up as being associated with the disease, then that is telling you that there's a problem with your study. Now, why? It is because we think that that's an extremely unlikely thing. So there's a prior that is coming in from biology, which tells us it's unlikely that every position in the genome is associated with the disease. And so then the way you explain it away is by bringing in hidden structure like Ancestry.
What were the different approaches that you may have chosen to reduce the feature set, or if the feature set was ever reduced in the first place?
Right, so that's a great question. So you're typically looking at 1 million features, and we try to use all of them as much as possible. And the reason is each of those features has very little signal. So each feature by itself doesn't tell you much about the ancestry. So it's by aggregating across all of these features that you're able to say whether somebody has ancestry X versus Y. So to the extent possible, we use all of the available information to be able to infer these quantities.
Yeah, okay. My question is, when you're gathering hospital records to infer the solution to your problem, so in that case, is everything going from the hospital record? Means there must be something that is having a higher priority. Means my question is basic data filtering. You can't put, for example, something like hair color, something like eyes color. So you can just put everything. But
Right.
why is it important, and what should be done to filter our data set? Is my question.
Yeah, that's a great question. So it turns out the actual raw data that comes from these medical records is huge. So the original medical records, it has about 50,000 tables, not features, tables. And those have all kinds of information. For example, when the nurse was checking you, did they click on certain boxes, which may have no relevance to clinical outcome. And this is where having clinical collaborators who can take the raw data and figure out which of these is a reasonable set to work with is extremely important. In fact, these are clinical collaborators who are quite sophisticated computationally. So they actually built databases which narrowed down these 10,000 tables to one table which had hundreds of features, which is the place where we can enter and say, amongst these features, which ones are most likely to be important. So in all of these studies, it's important to have this dialogue going on with the domain experts to figure out what is a good starting point for machine learning. And once we have these systems working, we actually want to go back and see if there's something important that they left out. So now we actually want to go back to the raw data and not use the initial filtering and see how much additional accuracy we can gain.
So I want to talk about, you talked about that the disease which you identify has a high correlation with the ancestry, right? That's what you figured out. That given a certain composition of ancestry, it will have a high. So agreed that it has a high correlation, but what if you control for that population? If you keep the ancestry composition constant, do you find other variables which help in predicting any risk?
Right, so in that specific study, you find additional mutations. So after you look at a person's local ancestry, there are mutations that are sitting on that local ancestry that actually lead to increased disease risk. I mean, that's what the clinicians are most interested in. They want to actually figure out, this is the mutation that we should be targeting for developing a drug. And there have been instances where there's an ancestry effect, but there was no mutation effect, whereas what we actually care about is actually finding a mutation. So in the example that I showed, it turns out that there's actually a SNP effect that actually explains some of this differences in disease prevalence.
Okay. My question is, you are looking at ancestry as one of the parameters to predict disease. Is it only about ancestry, or is it more about lifestyle, healthy lifestyle or not a healthy lifestyle?
That's an extremely important question, which is how much of any of this is genetics versus environment. So there's a huge debate in this field in general about trying to understand what we call heritability. How much of your type 2 diabetes risk is genes versus the environment. And so we and others have been doing a lot of work on trying to get at that. And currently, it's a quantitative question. It seems like for some diseases, they're highly genetic. Breast cancer is an example of one of those. Others, they don't seem to be highly genetic. We don't know if that's because we don't have the right sample sizes, we haven't collected the right set of mutations. So that is still an open question.
Hello, Professor. Hi, at the back. So my question is regarding the admixture model. You mentioned population 1 and population 2, and took some examples like North American, European, but these are very smoothed out, clean populations. But how does the model transfer to India, where there are thousands of endogamous groups? Like, is it relevant to India?
Yeah, that is an extremely interesting, complicated question. There are multiple differences. So one is, so in all of this, again, this goes back to data sets. A lot of what we know about all of these other populations is because we have huge genomic data sets in Europe and the America. India, we haven't had these large data sets to actually understand what's going on. So I'm fairly sure that there's a very complicated picture about ancestry within India as well. There have been a few studies, but going down to the detail of how this varies in region, north versus south, east versus west, yeah, it's interesting. We just don't have the resolution to actually answer some of those questions.
Thank you, Sriram.
It's on? Okay, yeah. Thanks a lot.